Which of the given statements is incorrect about microarray or microchip analysis? A. It is a new technology in which all of the genes of an organism are represented by oligonucleotide sequences spread out in an 80x80 array on microscope slides. B. The oligonucleotide sequences cannot be synthesized directly on the slide. C. The oligonucleotides are collectively hybridized to a labeled cDNA library prepared by reverse transcribing mRNA from cells. D. The amount of label binding to each oligonucleotide spot reflects the amount of mRNA in the cell. The correct answer is the oligonucleotide sequences cannot be synthesized directly on the slide. Which of the given statements is incorrect about microarray analysis? A. It is designed to detect global changes in transcription in a genome. B. It provides information about the levels of protein products of the genes. C. The proteins are first separated in a column on the basis of size and then across a second dimension on a slab on the basis of charge. D. Labeled protein samples may also be extracted from treated cells and separated by two-dimensional gel electrophoresis. The correct answer is it provides information about the levels of protein products of the genes. In cluster analysis of microarray data, a node is created between the scoring pair and the gene expressed. Profiles of these two genes are averaged and the joint elements are weighted by the of elements they contain. A. Lowest frequency. B. Average sequence. C. Lowest number. D. Highest number. The correct answer is highest number. In cluster analysis of microarray data for n genes, the process is repeated times until a single element remains. A. N2. B. N. C. N1. D. N4. The correct answer is N1. Which of the given statements is incorrect about global gene regulation? A. One way to obtain useful information about a genome is to determine which genes are induced or repressed in response to a phase of the cell cycle. B. Sets of a gene whose expression rises and falls under the same condition are likely to have a related function. C. Sets of a gene whose expression rises and falls under the same condition are likely to have dissimilar functions. D. Cell cycle is a developmental phase or a response to the environment. The correct answer is sets of a gene whose expression rises and falls under the same condition are likely to have dissimilar functions. In cluster analysis of microarray data, if Xi is the log odds value for gene X at time I, then for two genes X and Y and N observations a similarity score is calculated. SXY is also known as the Pearson correlation coefficient. X-offset and Y-offset can be the mean of the observations on X or Y respectively, in which case is the standard deviation, or else X-offset and Y-offset can be set to 0 when a reference state is used. Which of the following best represents it? A. SXY frac 1N2 1N2 I1N. Xi X-offset Y offset XQY. B. SXY frac 1N 1N I1N. Xi X-offset Y offset XQY. C. SXY frac 1N 1 1N 1 I1N. Xi X-offset Y offset XQY. D. SXY frac 1N 1N I1N2. Xi X-offset Y offset XQY. The correct answer is SXY frac 1N 1N I1N. Xi X-offset Y offset XQY. In self-organizing maps, a choice is made of a number of clusters by which to organize the data. A. True. B. False. C. D. The correct answer is true. Once a set of genes that are co-regulated has been found, the promoter regions of these genes may be analyzed for conserved patterns that represent sites of interaction with specific transcription factors. A. True. B. False. C, D. The correct answer is true. SVMs support vector machines are a binary classification method to discriminate one set of data points from another. A true, B false, C, D. The correct answer is true. The hierarchical clustering method generates a similarity score SXY for all gene combinations, places the scores in a matrix, joins those genes that have the highest score, and then continues to join progressively less similar pairs. A true, B false, C, D. The correct answer is true. In SVMs support vector machines, data points are logged, transformed, and normalized as in method A, where for n observations of a gene I the log transform she of the expression level A and reference level R is A she frac log EIRISQRT sum J1n log Z2 EJRJ log EIRIJ1n log Z2 EJRJ. BXJ frac log EJRISQRT sum J1n log Z EJRJ log EJRIJ1n log Z EJRJ. C she frac log EIRISQRT sum J1n1 log Z EJRJ log EIRIJ1n1 log Z EJRJ. D she frac log EIRISQRT sum J1n log Z EJRJ. The correct answer is she frac log EIRISQRT sum J1n log Z2 EJRJ log EIRIJ1n log Z2 EJRJ.